Hello, everyone. It's a pleasure to be here today. I am professor from the Department of Biochemistry and Molecular Biology at University Federal of Viçosa. I'm going to talk about the development of new biotechnology approach to combat bacterial resistance. The University Federal of Viçosa is located in Viçosa, a city of Minas Gerais state, localized in the southwest of Brazil. In this university, I am the head of Laboratory of Synthetic Biology and System Biology Modeling that develops technologies to identify and eliminate pathogens and chemical contaminants from human, animal, and plants, include the development of rapid test kits, bioplastic filters, characterization and optimization of new enzymes and new antibiotics, and pathogen control by genome edition using CRISPR-Cas9 approach. In my lab, we work with bioinformatics methodologies, include artificial intelligence and machine learning to identify potential targets in omics data. We also have expertise in synthetic and molecular biology techniques to validate the targets identified in the bioinformatic analysis and development of new products. In this talk, I will show an example of a project associated to development of three new disruptive technologies to control antibiotics-resistant pathogenic bacteria. Bacterial resistance is a serious and urgent public health problem that affects not only human health, but also the livestock and agriculture sector. In my lab, we have been working to develop technology that hindered or blocked the environmental spread of resistant genes, new approaches and devices for detecting infection by antibiotic-resistant pathogens, and new genome editing resistant reversal technologies as an alternative method to complete treatment these infections. Different human activities release antibiotics molecules from the environment, and the current wastewater treatment is not efficient to eliminate the mass of antibiotic compounds that exert selective pressure that accelerate the evolution and dissemination of antibiotic-resistant pathogens. The use of a filter that removes antibiotics of effluent may contribute to reduce the dissemination of those antibiotic-resistant bacteria. Our strategy is the use of aptamers to remove antibiotics. Aptamers are DNA RNA molecules that binding with high affinity and specificity to different targets. We use DNA aptamers in my lab because they are more stable than RNA sequences. The main advantages of aptamers compared to other approaches such as antibiotics is the price. Aptamers have low cost because they are produced by chemical synthesis, and aptamers are easily linked to cellulose membranes. The classical identification of potential aptamer sequence is performed by SELEX method, in which the target which could be a molecule such as an antibiotic, a resistance protein, or a cell is immobilized and brought into contact with a random DNA library. After washing to remove unbound oligonucleotides, DNA probes that specifically bound to the target are eluted and used in additional negative selection step to remove probes that bind into other targets, resulting in cross-reactivity. Specific probes are amplified by error-prone PCR to increase the variability of sequences that already have a select affinity for the target. This cycle is performed several times, resulting in probes with great specificity and affinity at each cycle. After the last cycle, the sequence obtained are cloned and sequenced. Due to the high cost and long term to obtain the aptamer sequence by the SELEX method, We have developed a selection method based on structural bioinformatics and machine learning that simulated a target in a defined environment that is surrounded by endonucleotides. The algorithm selected the nucleotides with the greatest potential of the interaction with the targets and join these nucleotides to produce an aptamer sequence. Although the program is hit rate it's 60%, the rapid synthesis and low cost of oligonucleotides result in a much more efficient approach to identifying aptamers than the SELEX method. This computational approach was used to identify potential probes to binding to antibiotics. The filter prototype is produced using cellulose extracted directly from plant waste, as for example, from coffee industry. So we use potential agriculture waste that could contaminate the environment to produce a new product to remove other environment contaminants. After the polymerization of the cellulose bioplastic, the aptamers are linked by chemical modification. You can see a prototype of a complete filter using my lab to validate the potential to remove antibiotics from wastewaters. Nowadays, our filter is able to remove 3 families of antibiotics and has a low production cost. We are also working in a similar filter to remove coronavirus from air, drinking water, and wastewater too. With antibiotics bound, the filter can become saturated and no longer be able to bind the molecules after approximately 1 year. The filter is able to offer regeneration using an acid solution that produces a concentration antibiotic solution that is easier to be eliminated or reused. The filter can go through 3 regeneration process without losing half of its removing capacity. The second technology is the use of the same aptamers in new technologies for rapid on-site detection of resistant bacteria in human samples. The clinical identification of bacterial resistance is mainly performed by on-disk diffusion or with test methods that can take days to generate results, or by evaluation of a patient response to different drugs by a trial and patient response. Such approaches are far to being an ideal point of care diagnostic test that needs to be safe to generate the reliability results, fast, allows using any environmental that is necessary, and be easy to manipulate it and execute it. Therefore, we are developing diagnostic approaches that have these advantages for detecting bacterial resistance. In this project, we work with E. coli bacterium that causes more than a third of deaths associated with antibiotic resistance. And specifically, we work on development of a technology to identify the beta-lactamase enzyme encoding by the blighting genes. One of the enzymes most frequently associated with clinical resistance to beta-lactam antibiotics. First, we use the bioinformatics approach to identify aptamers that bind specifically and with high affinity to blighting enzymes. The aptamer was used in a detection system based on gold nanoparticles. Aptamers has a high affinity for gold particles and cover these particles. In the absence of the target, the covering of the aptamers prevents the nanoparticles from aggregating, and the solution remains red. In the presence of the blotting enzymes, The aptamers release the gold nanoparticles and interact more strongly with the targets, allowing the nanoparticles to aggregate, making the solution blue. This reaction is very, very fast, and the result is obtained within a few minutes of reactions. As a picture is worth 1,000 words, I brought a video to demonstrate the detection efficiency of these technologies. Please note that we have a tube on the right containing the negative solution in the absence of resistant bacteria, and the color red is present. Here is the negative solution. And after adding a sample containing resistant bacteria, in a short time, the color is changed to blue, and this body can be quantified and is proportional to the amount of resistant bacteria in the sample. Compare the colors blue detect the presence of resistant bacteria. Now you are now integrating our probes into fiber opticals based electronic device to produce a specific device for detection and quantification of resistant factors coupled with identification pathogen species in human samples that could be used in hospital and assist in deciding which antibiotic to use in determined human. Another project is the use of genome edition approach to reverse the antibiotic bacteria resistance as efficient alternative to resistant bacteria treatment. Due to the difficult discovery and development of new antimicrobial molecules, including the long time necessary and expensive associated cost, alternative treatment are urgently needed. An example is the use of CRISPR-Cas9 silence methodology has a potential to eliminate the expression of resistant genes, making the bacteria susceptible to antibiotic again. The first step is the identification of potential guide RNAs that target the selected resistance genes. For this, we use several approaches like machine learning to identify the best RNA sequence, compare several resistant gene sequences obtained from public database. After selections, the oligonucleotide is synthesized and cloned in a specific DNA vector to be able to reverse the resistance in the bacteria. Susceptible E. coli strain, commonly used in the laboratory, include in experimental groups, this BL21 strain were transformed with the vector containing the CRISPR system to access that the vector did not directly kill the bacteria, and you use it with resistant bacteria. BL21 transformed with two vectors. One vector contained green fluorescence to access viability, and another vector contained the blatant resistant gene and had fluorescence. To access the potential to reverse resistance, the resistant induced BL21 was transformed with the vector containing the CRISPR system. We confirmed that the system affects specifically the resistant genes using fluorescent vectors. You can see in the figure represented the result of the growth assessment with each experimental group in the absence or presence of ampicillin. Absence of ampicillin is the black dots, and absence of the presence of ampicillin is the gray dots, sorry, and the absence of ampicillin is represented by the black dots. The arrow represents in the time of addiction of ampicillin, 6 hours after the start of cultivation. In the graph, in the figure A, represent the susceptible bacteria, which reduced growth after this, when the antibiotics is added in the cultivation medium. In the figure B, the bacteria contain only the vector crisp showed that it does not directly affect the physiology of the bacteria, and the same growth profile is observed without the presence of the vectors. When the bacteria resistance was induced, there was no difference in growth when comparing the absence and the presence of the antibiotic. Using the resistant bacteria, when the CRISPR vector was inserted, antibiotic sensitivity returned with a growth profile similar to that of a susceptible bacteria, indicating that the CRISPR system was efficient in silence the resistance genes. We confirmed that the system affects specifically the resistant genes using fluorescent vectors as a control. There are a specific reduction of red fluorescence, suggesting that the CRISPR system does affect the bacterial viability in green fluorescence. But silence the resistant genes lead to plasmid background. Thus, in addition to silence the genes, the technology contributes to reduce the horizontal transfer speed of resistant genes by eliminating the plasmid that host these resistant genes. A sequence and comparison of the wild and Egyptian resistant genes demonstrated that the CRISPR system induced a premature stop codon, silence the prevent complete protein synthesis, resulting in a smaller protein with no enzymatic. The potential of system based on CRISPR-Cas9 was validated in clinical isolated with natural resistance to ampicillin and the other antibiotics by this diffusion method. With the reversal of resistance is proven by increase of inhibition, halo, after contact of the bacteria with antibiotic, for example, for ampicillin antibiotic. In different species of E. coli and Enterobacter cocci, the resistant gene silence and plasmid elimination by CRISPR system was efficient to increase the sensitivity for different antibiotics. The challenge of this technology is the delivery of the CRISPR-Cas9 system to pathogens in infected hosts. Our proposal is the use of phage to deliver the system during the high specificity to phage to inject its genetic material into bacterial cell. The CRISPR system is inserted in the phage genome, and this phage to deliver the system and reverse the resistance was validated using Galeria model. For example, to ampicillin resistance, the CRISPR system delivered by the phage demonstrated by the headline increased the survival of infected larvae from 25% to 70% compared to untreated samples in yellow lines. Challenges for development this technology include the high selectivity of phage to bacteria, which would require the use of phage cocktails to apply to different pathogenic strains or species. The validation of the absence of impact on the host natural microbiota and the absence of negative effects on the host health and environmental safety, as well as the development of control and monitoring methods for these genetically modified phages. My lab works in very collaborative environment, and I'd like to thank all the people and institutions involved in development of the projects. I also like to thank the public agents and private companies by the financial support. Thank you so much for your attention.